So we're going to talk about plasmid, a topic under microbiology. So the definition is it's an extra chromosomal double-stranded DNA molecule location in the free state in the cytoplasm of the bacteria. So in the diagram, as we can observe that here is the blue colored bacterial chromosomal DNA, and the other two, the red color, are plasmid, which are lying freely in the free state in the cytoplasm of the bacterial cell, and its definition is extra chromosomal double-stranded circular DNA molecule. Now, some of the characteristic of plasmid are that it is a non-essential, so bacteria may gain or lose plasmid during its lifetime. Numbers, it could be single or multiple in number. Independent replication, it behaves as replicons and has origin of replication and other genes responsible for its independent replication. Episome is called when it integrates with the chromosomal DNA of bacteria. So here we can appreciate episome, where the blue one is a bacterial chromosomal DNA and red is RNA. So when red one is a plasmid, and when plasmid gets integrated in the chromosomal DNA, it is referred to as episome. The curing, elimination of plasmid from the bacteria, is referred to as a curing. So some of the characteristic of the plasmid are non-essential. Some bacteria may can gain or lose it. Numbers could be multiple or single. Independent replication, so it behaves as replicons with origin of replication and other genes. Episome, when it gets integrated with the chromosomal DNA, and curing, elimination of plasmid from the bacteria. Now, classification of DNA is subdivided into three topics based on the ability to perform conjugation. Second is based on compatibility between plasmids, and third is based on function. Total 5 functions are there. So first, based on ability to perform conjugation, there are two type of plasmid: conjugative plasmid and non-conjugative plasmid. So ability of to transport themselves to other bacteria by means of conjugation, which is self-transmissible conjugative plasmid, is referred to as a conjugative plasmid. And non-conjugative plasmid is also known as non-transmissible plasmid, which cannot transfer themselves to other bacteria by the means of conjugation. Okay, so and now we'll move on to the second one, based on compatibility between plasmid. The plasmid compatible bhi hote and it can be incompatible. So compatible plasmid, different plasmid can exist in the same bacterial cell only if they are compatible. So though plasmid, if compatible ho, then they can survive in a single cell. But what if they are not compatible? Then it is referred to as incompatible. So either they compete with each other and one of them is lost. That's how they exist. Now, based on the function, there are total 5 function is there. Fertility or F plasmid. तो उसके पास कौन सा जीन होगा? Fertility or F plasmid. What kind of gene do they have? The gene that they will carry is TRA genes that codes for expression of the sex pili, helps in the bacterial conjugation. Okay. Then resistance R plasmid have genes that codes for various antibiotic resistance. बहुत सारी antibiotic के resistance चाहिए, then we will have resistance plasmid that will code for that. Col plasmid genes code for bacteriocin, antibiotic like substance produced by bacteria that kills other bacteria. Okay, that is col plasmid. And virulence plasmid, obviously virulence means pathogenesis कुछ करेंगे, so codes for virulence factor and toxins, therefore the bacterial pathogenesis will occur. Some of the example is heat labile, heat stable toxin of E. coli, siderophore production, adherence antigen K88 plasmic of E. coli. Then metabolic plasmid, they will perform some of the metabolic activities. So helps in metabolic activities such as digestion of unusual substances such as toluene or camphor, urease synthesis, nitrogen fixation. Okay, these are the 5 types of plasmid function. So based on the function, fertility, resistance, then we have is col plasmid, virulence, and metabolic. Now plasmid as vector. So ability to transfer DNA from one cell to other is referred to as a ability to transfer DNA from one cell to other, we will call it as a vector. So plasmid is an important vector in genetic engineering. So in the plasmid we talked about its definition, which is extra chromosomal double-stranded DNA, location in free state in the cytoplasm of bacteria, and some of the characteristic of the plasmid, such as it is non-essential. It's we talked about its number, that it could be single or multiple. Then we talked about its independent replication, episome, and curing. And classification of plasmid was based on the ability to perform conjugation first, second based on the compatibility of the plasmid, and third one was based on the function of the plasmid. And the last topic was plasmid as vector. So it is a very important component or a vector in a genetic engineering